Okay, video recording. No. Okay, so let's get started. It's no different. Try not to be any different then, and I can change things. Okay, no big deal. Okay, so protein synthesis. So let's talk first. Let's always keep our frame of reference here. Okay, so that's important. So, all right. So think about the cell cycle for a second. G1, S, G2, mitosis, cytokinesis. During G1, during G1, what typically happens during G1? What's being prepared? Well, okay. What's being prepared during G1? Okay, so not necessarily. Okay, so during G1, if you think about G1, it's just like we said, it's just the cell living, right? It's just, in other words, it's like if you're a pancreas cell and your job is to make insulin and glucagon and hormones, that's what you do, right? You eat a meal, okay? I secrete some insulin to help my body uptake nutrients from the meal. That's what pancreas cells do, or glucagon, whatever the case may be. So that's G1. It's like once the cell hits that checkpoint during the end of G1 where it says, uh-oh, something's happened, I need to divide. It may be the cell's getting old. It may be that something's damage has happened, something. The DNA needs to replicate. Then it gets to S phase, right? Replicate your DNA, make a copy. And now the cell will divide. G2, grow a little bit more, prepare for mitosis. G2, mitosis, actually divide the nucleus, mitosis, and then divide the cytoplasm and cytokinesis. And now you're back to G1. So the point of saying that is during proteins, when we're kind of thinking protein synthesis, we're kind of thinking G1. Like that's where that main amount of protein synthesis has happened, where the cell is just doing what it does. Doesn't mean that proteins aren't made during DNA replication, but probably not many, because think about it. The DNA is being replicated. It can't, because the DNA is the code, right? So if the DNA is being replicated, it's probably not a good time to be trying to use the DNA to make messenger RNA, which we'll talk about, to make proteins. In G2, there is some proteins being made. In mitosis, there's probably not hardly any proteins made, because besides the ones that are already there. Okay, so that's what we wanted to kind of keep in mind, is we're in G1 here for the most part. That's what we're thinking about, is G1 in the cell cycle. Okay, so I'll be quiet and let you guys write here some of this basic stuff. Just kind of get some definitions. What is a polypeptide? And that's the main idea to get here. You don't really, for our purposes, this picture is not in your notes, is it? No. Okay. So we do need to know, I want you to write this, though. I need to write on the side, and I'll give you plenty of time here. The primary structure is the amino acid sequence. The secondary structure are structures like alpha helixes or beta pleated sheets or just beta sheets. And then you have this tertiary structure. That means tertiary means 3. And then quaternary, like a quad. You know a quad, how many wheels does a quad have? 4. So you think quaternary is the 4, right? So, but take a minute there and kind of write that in. And I think we have another page in our notes that actually has this on it. So it may not be necessary to write it right now. We'll get it later. But polypeptide. Proteins are one of my most favorite things, though, guys. This is something that's such an integral part to biology and science. It's just so important. And if you have kind of a basic understanding of this, it gives you some ideas to, you just, again, like we always say, you can test what you know against what you hear. And then it will allow you to kind of process things a little differently, right? You hear something, and you're like, wait a minute, that doesn't agree with what I know to be true about proteins. And then you can either, well, maybe I need to look some more stuff up. Or maybe what I'm hearing is not correct. And you can kind of say, wait a minute. So, as you're finishing writing here, I want you to think about these structures for a second. So, first off, let's read this polypeptide chain. A polypeptide, what does poly mean? No. Poly means... What'd you say the second thing? Multiple. Poly means many, right? Mono means one, right? You said mono, there's a monopeptide, the one peptide, but poly means many peptides, okay? So it's a chain of amino acids can bind to others and fold into a protein. So a protein is not until it's done. You call it a protein. So we call it in the basic sense a polypeptide, yes? We'll turn in just a second. Just keep it right there and we'll turn in. Yep. So let's think about this wall behind me. Got this big giant wall. So that's a good kind of analogy. A polypeptide. A polypeptide is a bunch of amino acids, right? Remember the monomer for proteins. It's a polypeptide. It's an amino acid. So this wall behind me, what would you say? If this wall is the protein, what are the cinder blocks like? Think through relative to what we're talking about up here, and this word right here. If the wall, the entire wall is the protein, what's the cinder blocks? Yes, they're amino acids. So amino acids are building blocks. You put a bunch of them together, you make a protein, which could be the wall. Okay? Now, what starts to happen for a protein, if we just talk briefly through these structures, and not in much detail, guys. The secondary structure, proteins, they start to fold up on themselves. Once you start to put a bunch of amino acids together, they start to interact with each other, and they start to fold up. And proteins start to have shapes that define their function. And that's why, have you ever heard, like you can, why if you were to use something hot, it would kill a protein or something like that, or kill things that are hot? You know what I'm kind of talking about there? That doesn't make much sense. But if you were to heat things up, the reason it like neutralizes bacteria and things like that, or one of the reasons, is because when you heat these proteins up, you change their shape. You change their shape and they no longer function. It's like when you heat a piece of metal up and it starts to change its shape. Well, it doesn't function the same as it used to because the shape is different. The same thing with these proteins. If you heat them up or you change other things about them, you change their shape and they don't work anymore. Okay? Okay. More about this later. The main thing to get here is the process of reading the instructions in DNA to make a polypeptide. And polypeptide is a chain of amino acids. We'll continue to make sense of this on the next slide. Okay. All right, I'm going to give you a few minutes here, guys. This is a really good image to make all of this make sense. We're going to spend a lot of time on this slide. But so go ahead and write in this right here. Really make sure you get this. This is some good, important stuff, and we'll talk about it in just a second. This picture is in your notes, right?
Yes.
Okay, good. That's good. This slide, as you're finishing up writing here, this slide is called the central dogma of genetics. Like, what the heck does that mean? The central dogma. Just kind of think about it as like the big idea of genetics. And so this is like the deal, you know. Talked about different ideas and different things to learn. Man, if you can learn this part, how do you get from DNA to protein? That's the deal, man. That is the deal. There's so much in terms of what especially now with, with the manufacturing of vaccines and all of the terminology that's on the news and stuff, man, if you can understand some of this, you're going to have a much deeper understanding of the things that you read and hear. And like I said, you can really start to put a few puzzle pieces together and start to make sense of a few things. That's why this is really important. It's not like you got to be a biochemist, but you got to have a basic understanding. So this picture is beautiful, guys. So let's take a look at this picture for a second. So what in the world are we looking at right here? What is this whole thing? What is this picture of? The picture of it's a picture of a cell. Exactly right. Very good. So we're missing some stuff here, but let's just make sure. What does this circle right here represent? The nucleus. Exactly right. All right. So obviously in the nucleus we've got the DNA. This is a little piece of DNA they're showing. And now it's just labeling out these processes. So what I'll ask you on a test is something like, hey, where does transcription take place? And you'd say the nucleus. I would say, where does translation take place? In what's the fluid field? The cytoplasm. Beautiful, yes. And we probably say, really and truly, probably most often in the rough ER. Really and truly, because remember, the rough ER is attached to the nucleus. The messenger RNA, through transcription, is going to be made in the nucleus. It can leave through a nuclear pore into the rough ER, because messenger RNA doesn't like to just hang out in a cell. It'll be chopped up by enzymes. So it's delicate. And that's one of the things, because it's single-stranded, doesn't have two sides. It's not very stable. That's one reason why we don't store our genetic information. You may say, a good question is, why do we even have DNA? Why isn't it just all encoded in messenger RNA? Why don't we have just tons of chunks of messenger RNA? But one of the reasons is because it's not stable. It gets degraded easily. So it's going to leave, go into the rough ER, and then translation can happen. Okay, going to change that language, so to speak. Okay, and that's where we get made into a protein. So this green glob right here is a ribosome. Okay, and we have a lot of stuff going on right here where we have these things called transfer RNA. We're going to look at this on the next slide. We have messenger RNA. We have ribosomal RNA that actually helps make up this big green glob right here. So the ribosome is made of RNA and protein. But the big thing to get here is this. Where are these two things located? And this whole process. The process of making DNA, the process of getting DNA into RNA is called transcription. The process of getting RNA to protein is called translation. Okay? So that's the big deal, DNA is in the nucleus. Does DNA ever leave the nucleus? No. It can't, right? It doesn't ever leave the nucleus. So, and it's another thing we should think about, right? So it makes sense. Are there any ribosomes in the nucleus? No, there's not. So it begs the question. It's like, okay, well, if ribosomes make protein and they're not in the nucleus, well, how do we get any protein made? Because DNA is in the nucleus. So we have an intermediate step, RNA, which can leave the nucleus and go to the cytoplasm or the rough ER to be made into protein. Okay? Awesome, guys. Okay, so let's do this one. There's a lot to write here, but just go ahead, take a second, write these words in, and we don't want to get bogged down in the details so much right now. We'll get the details later. Just try to keep the big picture in mind. A lot of terminology: messenger RNA, transfer RNA, ribosomal RNA. Yes, yes. Because you do want to be able, this could be test questions. We're going to ask you, hey, what's a messenger RNA? What's a transfer RNA? What's a ribosomal RNA? Those type of things. Yeah, definitely want to know these. We'll watch a video of this in action, like a simulation of it, which to me, I like to see the the pictures, kind of visual. And so maybe that'll work for you too. As you can kind of see it in action, it helps. You can put that puzzle together, and you might get to kind of visualize the whole process. That helps me. So I know some of you are still finishing up writing. What do you notice? What are these things right here? What are those called? And here. No. Okay. Remember, think about it in terms of drawing our nucleotide. You have the phosphate, you have the sugar, and the nitrogen base. These are all nitrogen bases. Okay? They're all nitrogen bases. That's all it is. So, I thought we got it at 43. Why do I always do this? This is fourth period. I'm sorry. End. Very good. I don't know why I always do this. Okay. Good, guys. Put up your stuff. Thank you. Great job. Great job.